Good afternoon, everyone. Happy Friday, happy Valentine's Day, and welcome to the last webinar of our Winter Water Talk series for this season. I'm Paul Skowinski. I'm the statewide educator for the Citizen Lake Monitoring Network and part of the Extension Lakes team at UW-Stevens Point, and also joined by my colleagues Katie Bradford and Emily Heald from the Water Action Volunteers Program. If you missed any of our previous water talks from this season, here's a schedule from this season. If you see anything of interest to you, you can find recordings of these webinars on our YouTube channels, either the Extension Lakes channel or the Water Action Volunteers channel. And there are many past webinars on there as well. So you can go and watch them or rewatch them or share them with somebody else. If you have a question today, you'll use the Q&A button on the bottom of the screen. Sorry, someone's calling me on Teams right now. Use the Q&A button at the bottom. You can enter a question at any time during the presentation today, but we are going to hold most of the questions until the end. And without any further delay, Chris Taylor is here from the Illinois Natural History Survey. Chris is a semi-retired aquatic biologist and emeritus curator of fishes and crustaceans at the Natural History Survey at the University of Illinois Urbana-Champaign. He's worked at the survey for 31 years, where he conducts research on the systematics, ecology, and conservation of non-game fishes and crayfishes, and oversaw the growth and care of the survey museum collections of fishes and crustaceans. Chris has published over 90 peer-reviewed papers, including 4 books on the identification and ecology of crayfishes in the Midwest, the fishes of Illinois, and crayfishes of Kentucky and Alabama. Chris continues to stay active in the conservation of native biodiversity and freshwater habitats. So Chris, I'm going to stop my share here and you have the remainder of the hour for your presentation.
Okay, thank you, Paul, and thanks for everybody for joining. Let's see if I get this right. Are we good?
Looks good.
Okay, all right, great. Well, thanks everybody for the opportunity to talk to you. I've got maybe about 42 or 3 slides to talk about some introductory material on biology and ecology of crayfishes. And then the last part, we'll talk about identification and which species occur where in Wisconsin. How do I advance the slides?
You should be able to click on the screen, or I think you can use your left and right arrows too.
We got it?
All right.
Okay. So first, we need to talk about what is a crayfish or crawfish or crawdad. Hopefully that will head off one question I usually get. What's a crayfish and what's a crawfish? One and the same, all just locally used names for the same critter. But kind of a formal definition of what is a crayfish. It's an aquatic or semi-aquatic arthropod that respires with gills, has 10 pairs of non-feeding appendages, hence the name. It belongs in the order Decapoda. So there's where the deca comes from 10 pairs. And it has direct development. And by that, crayfish's closest non-crayfish relative are marine lobsters. If you think of something like a Maine lobster or North Atlantic lobster, it looks a lot like a giant crayfish. The difference being that marine lobsters have a larval stage, like crayfish have direct development. Their eggs hatch into tiny little crayfish, and those tiny crayfish just grow to become adults. All right, so just a little overview on where you find crayfishes in the world. There are 3 different families. Family is that taxonomic organizational unit. Families are Cambaridae, Astacidae, and Parastaccidae. You can see from the different shading on this map that crayfishes occur across most of North America in the family Cambaridae, a little bit in East Asia. The Astacid family occurs in the Pacific Northwest and Western Europe, and then the Parastaccidae are known as the Southern Hemisphere crayfishes from New Guinea, Australia, New Zealand, over to temperate regions, small temperate regions in South America. One lesser known fact is also the diversity of crayfishes in the world. There are about 620 known crayfish species in the world, and an overwhelming majority of those are found in North America. If you look at this illustration with its color shading, the darker color of blues indicate highest levels of diversity and where you find that diversity. And you'll notice right away that the southeastern United States has the highest diversity of crayfish in the world. I can't tell you the polygon shapes here that are shaded off the top of my head, but in any one of these regions here in the southeast, you can easily find greater than 50 crayfish species. So we're in a good spot in terms of the US for crayfish diversity. In terms of other places in the world, kind of a secondary area of biodiversity is over in the southeastern portion of Australia. All right, unfortunately for Wisconsin in the cold upper Midwest, diversity drops off precipitously. There are 9 total species of crayfish that have been documented in Wisconsin waters. 7 of those are native, 2 of those are exotic or invasive, and we'll come back to talk about those in a few latter slides. Just putting things in perspective, Wisconsin is not the worst off. Montana has only 3 native species, so as you move north and west, diversity of crayfishes drops way off. This is an example of that high biodiversity in the southeast. The state of Alabama has 97 native crayfish species. All right, so a little bit of ecology of crayfishes. Crayfishes are found in just about any type of aquatic habitat, most certainly in the eastern US. Everything, any type of lotic or lentic, flowing versus non-flowing habitats. We kind of break them down into several different categories. One category is the epigean or surface dwelling species, and within that, anything from large navigable rivers here, as you see in the upper left, this is probably the Mississippi River in southern Illinois, to very small headwater streams or creeks, large impoundments. You see in the lower right, and as an example of a lentic system, sloughs and swamps and backwater areas. Crayfishes have colonized and do quite well in all those types of habitats. If it's wet or even semi-wet, you will generally find crayfishes, especially when you get to the eastern US and the southeastern. Now, I said semi-wet because then there are what we call the hypogean species, or those that occur in subsurface waters. And those can be 2 different types. Either start with cave dwellers here on the bottom, as you'll see. There's about a dozen, there's probably 15 species now that occur entirely in caves. They are true troglodytic organisms. They lack pigment, as you see here on the lower right. They have small, greatly reduced, non-pigmented eyes, and you will only find them in that subsurface streams that flow through caves. These things do not see the light of day. The other type of subsurface water dwelling species are what we call burrowers. If you notice this photo on the left, most of you have probably seen wet roadside ditches or backwater areas that are only seasonally flooded. And then as that water dries up, the soil stays very, very moist. Burrowing crayfish have adapted the ability to dig down into that soil and survive quite well. I want to talk about burrowers versus surface dwellers or non-burrowers here in the next few slides. Okay, so of the non-cave dwelling species, those 15 species that you won't see outside of a cave, they can generally be divided into burrowers versus non-burrowers. The burrowers can sometimes be called primary or secondary burrowers. These are species that spend almost all of their life in deep subterranean burrows that they themselves have dug. As you can see from these center image illustrations, some of these burrows can be very, very complicated with multiple openings to the surface. usually one large central chamber in the middle. Um, the difference between a primary and a secondary burrower is a primary burrower does not have a connection to a permanent body of water. And down here, this arrow, I'm assuming you guys can see my cursor moving, correct?
Yes, we do.
Okay, so if you look down here, this is a secondary burrower. You see this tube coming over to this permanent body of water. That's what we call a secondary. But still, even secondary burrowers have very extensive burrowing systems. Some of you may have seen these mud pellet chimneys or mud pushed up to the surface. That's the opening to a crayfish burrow. Another example here of kind of a wet field that holds water, usually in the spring. And burrowers have moved in and colonized that habitat and spend most of the time underground in these burrow systems. One thing I want to point out here on this right illustration is this is the backside of a crayfish. And these two lines that form the margins of the gill chamber underneath that carapace. And you notice those lines come together and touch. And we're going to come back to that. It's really important when we start talking about identification of crayfish. The theory here is that since oxygen levels are usually quite a bit lower in these burrow systems and chambers, that the gills of burrowing crayfishes have become enlarged. And as they become enlarged, that portion of the body has forced these marginal lines that kind of demarcate the gill chamber underneath have gotten pushed together because the gill chamber has become enlarged to extract more oxygen. Frequently burrowing crayfish also have wider, more triangular shaped chelae or claws that they use kind of like a bulldozer to push that mud and dirt out. You notice on both illustrations, you'll see these squiggly lines. This denotes the water table. Burrowing crayfish will generally dig down and follow that water table as water starts to recede during drier times of the year, usually late summer and into the fall. If you notice in this top image, you see a chamber here in the center, and then this tunnel that's gone down a little bit deeper. What that means is as that water table in the late summer or fall starts to drop, the crayfish will follow it and dig another tunnel down to keep following that. These burrowing crayfishes can extract oxygen from the air with their gills, but their gills have to stay moist. And so they need to kind of track that water table a little bit to keep their gills moist. Down here, you'll notice I've got depths of some of these burrows have been recorded almost up to 3 meters. So these are very, very industrious organisms that have adapted the ability to move into these semi-aquatic habitats. Okay. As far as non-burrowers goes, this is where it gets a little bit confusing. I'll try to keep everyone on the straight and narrow here. Any crayfish can burrow to some extent. The true stream or lake dwellers have the ability to build a very shallow, you see over here on the lower right, a very shallow, simple burrow if they occur in a stream that usually dries out during periods of drought or in late summer, early fall again. And so that gives these normally stream-dwelling crayfish the ability to follow that water table as it starts to drop during drought periods and keep their gills moist if they're occurring down here. Sometimes you'll go into a stream bed that's mostly dry, and if you flip some of these rocks like you see here, you'll find a crayfish over here in the lower left that's gotten down into that soil moisture that's still present as that stream channel starts to drop. So we call these tertiary burrowers. Burrower is in the name, they're a tertiary burrower, but that means they only do it when they absolutely have to during times of drought. Okay. Shifting to kind of more diet and biology of crayfish. Check my timer to make sure I'm kind of on track here. Crayfishes forever were referred to as opportunistic omnivores, meaning they eat everything and anything that they came across. And that's still true to some extent. Um, there's all kinds of literature that shows the types of food that crayfish normally eat. They're both herbivores and carnivores. Lots of algae. They're known to scrape and consume algae in streams or lakes. You can see on the left here, both vascular and non-vascular plants, and then animals from 6 different phyla. Gastropods, for example, snails here, other insects, aquatic insects. They're known to consume fish. There's reports of them feeding on carcasses of both birds and mammals. So that lends support to the idea that they're opportunistic omnivores. Whatever's abundant and easy, that's what they'll feed on. But there has been some recent data in the last 15 years that shown that crayfishes seem to favor higher protein food sources as they age and become adults. So, in a sense, they're becoming primarily predaceous or carnivorous, feeding on an example. This is a Johnny darter here in the lower left, a species that's very common across Wisconsin, to something like a mayfly here on the right. And so they'll prefer to eat that higher protein food items, but then will fall back on plant matter if they have to. Okay, so normally as they're early stage younger individuals, they'll feed on whatever is abundant, but then as they become older and increase in size, they try to focus more on higher protein food sources. That's usually insects and fish. All right. So crayfish size, we'll talk a little bit about crayfish size. This is just a composite of several different species that are found in Missouri. It's a great example of the maximum body size for 5 different species here. And most crayfishes, when you talk about body size, you're basically talking about the length from the tip of what we call the rostrum, and we'll get into morphology here in a slide coming up, from the tip of the rostrum, which is the structure that is between their eyes to the end of their tail. They're normally in the 3-inch or below size range. There is one group of crayfish called the dwarf crayfishes that you see here in the middle of this image. Their maximum body size is no more than an inch. So a little bit of variation within North American species in terms of size, but in terms of crayfish in general around the world, they also do get quite large. The largest freshwater invertebrate in the world is a crayfish. This is the giant Tasmanian fresh, they call it a lobster in Australia. This is just a few images of how big a crayfish can get. This is that different southern hemisphere family, but it is still most definitely a crayfish. Okay, so said crayfishes eat almost everything at some point in their life. And that kind of leads into a discussion about crayfish's role in ecosystems. And here's a typical trophic food pyramid that I know all of you have seen somewhere in a biology course, starting with primary producers in an aquatic ecosystem, periphyton, algae, moving up to primary consumers at the second level. These are the insects or the things that scrape that algae, feed on that primary production, and then secondary consumers like smaller fish and crayfish, and then top level predatory piscivores in an ecosystem. It's probably pretty good for a Wisconsin lake with pike or muskie at the top of that food chain. Crayfishes are considered keystone species in many aquatic food webs. And the reason for that is if you notice the two red arrows here I've got on the right side of this pyramid. As I mentioned in the previous slide, crayfishes feed on both primary producers, plants or phytoplankton, and they also feed on kind of primary consumers, um, you know, snails and aquatic insects, for example. Crayfish are considered keystone species because they are taking that, especially the primary production, and packing that energy into a nice consumable piece of animal protein for top level predators. So they're that kind of quintessential link between primary producers in an ecosystem, vegetation, between that and top level predatory fishes or even mammals. Raccoons love to eat crayfish. Wading birds love to eat crayfish. So they're that key middle piece for taking plant protein and converting it to animal protein to move that energy up through a food web. There's been some studies that have shown crayfishes can account for up to 50% of invertebrate biomass in streams. So they're doing a lot of that energy. They're 50% of the biomass, say at this primary consumer level or secondary consumer level, they're moving an awful lot of that energy in a food web up to the next level. And another important system, we like to highlight this when we're talking about crayfish, is the importance to sport fishes. You know, we got the right food pyramid here for folks in Wisconsin. Sport fishes like smallmouth bass may consume up to 65% of their annual production in energy in crayfishes. So they're an incredibly important food source for those fishes that people love to fish for. Smallmouth bass is a great example, but certainly things like pike are in there. Some of the other sunfishes, walleye, are eating an awful lot of their energy is coming from crayfish. Okay, so we're going to transition to invasive species in the next slide, and this is kind of a good segue into that discussion because we're going to talk about invasive species, especially invasive crayfish species in Wisconsin. And we're talking about how important crayfish are to moving that energy up a food web. When an invasive crayfish becomes established and they displace native crayfish, it can create a disruption in some of that energy flow. Okay? So there are two invasive crayfish in Wisconsin, both the rusty crayfish and the red swamp crawfish. And you'll notice I have crawfish here. This is a common name that was assigned to the species in more southern locales. If you go to Louisiana or southern Alabama, they're not crayfish, they're crawfish. So someone used crawfish to give that species a common name. So not a typo. Okay, so Wisconsin is the hotbed for rusty crayfish. It was one of the first states that the rusty crayfish was introduced into, probably in the mid 1950s. And by the late 1980s, you'll see by the red dots on this map, were pretty widespread across the state. Rusty crayfish are found in both lakes and rivers. Anyone in the group here you'll notice up in the Vilas Forest County area up here in the north central part of the state, lots of dots. Those heavily fished lakes in those two counties were some of the first lakes that rusty crayfish were encountered. And they've had a huge impact on those lakes by reducing native species populations, in some cases, consuming an awful lot of that rooted vegetation, those macrophytes that are in those lakes. Those macrophytes are really important for juvenile fish shelter. Younger fish, smaller fish like to hide in that vegetation. It gives them some cover or some protection from larger fish that are out looking to eat them. So when the crayfish move in and chow down on that vegetation, there's a lot less cover for those poor fish. So the earliest research done on invasive species, invasive crayfish species, and their impacts on ecosystems were conducted up in the Trout Lake area here in northern Vilas County back in the early to mid 1980s. So Wisconsin, unfortunately, is the poster child for negative impacts that an invasive crayfish can have. They're also invasive in Iowa and Minnesota, and unfortunately, here in Illinois. This is just a map of how they spread in Illinois. The first place they were recorded was here in the Illinois River in 1974. By 1985, they'd shown up at various isolated locations. Some of this are probably spillover from Wisconsin individuals that came down the Fox River. And here's their range in 2015. So they spread like wildfire across the northern third of the state. All these other green dots on these two maps in Illinois are locations where someone had gone out and sampled crayfish. And we've got most of that data here at the Illinois Natural History Survey. So these locations with the green dots were sampled, and rusty crayfish were not present. So we know, we're very certain that until 1974, rusty crayfish were not present at an overwhelming majority of sites. The map here is covered with green dots. We've got great evidence that it has spread, unfortunately, and in a lot of these locations, it's the same thing. It's displacing our native crayfish species. A lot of these locations with red dots you can go to and you can no longer find native crayfish. All right, the other species that's invasive in Wisconsin is the red swamp crawfish. It is invasive around the world. If you look at the green dots on this map, this is another data source, iDigBio from National Science Foundation. Just looking at the United States, the red circle on the map here corresponds to their native range. They've been spread across the western U.S., the Pacific Northwest. They're on Maui, Spain, Portugal, several sites in Africa, in China, and Japan. They've been spread around the world, mostly for aquaculture reasons, for human consumption. So when you look at the number of dots now here in the U.S., they are literally everywhere. They did first appear in Wisconsin in the late 2010s, and they are still restricted to the extreme portion of southeastern portion of Wisconsin, and usually in ponds. And I've got, when we start going through species by species for identification, I have more detail on the range in Wisconsin. Okay, so this is the second one that's now established in the state. Okay. That's kind of the quick overview. If anyone has a quick question on biology or ecosystem function, happy to address it quickly before we move on. I'm going to talk a little bit about crayfish anatomy and then move on to identification.
Yeah, Chris, we did have one question that came in. Someone asked, why are there so many species of crayfish in the Southeast U.S. in particular compared to other parts of the world?
Yep, great. Multiple reasons. One, the southeastern U.S. was spared the last three glacial advances. The most recent glacial advance was 10 to 12,000 years ago here. That extent of that glacial advance was just south of Champaign-Urbana. So 12,000 years ago, everything was under 50 feet of ice. Okay, as those glaciers receded, aquatic taxa have slowly started to move back in. That southeastern region of the U.S. was never impacted by glaciation. So they've had hundreds of thousands, millions of years for speciation processes to progress. There's also an abundance of different habitat types from the southern Appalachians, where you get higher elevation, high gradient streams coming down from the Appalachian Mountains all the way down into, the backwater coastal plain area of what you typically would call a swamp or a bayou. And like a state like Alabama, you've got all those different habitat types between higher elevation streams all the way down to swamps and everything in between. The other reason is they get a lot of rain down there, and don't really have a severe winter, and so there's just a lot of, because of that moisture, a lot of aquatic habitat.
Thank you. That's great. We do have a variety of questions related to the invasive crayfish species.
We can circle back to that when I'll start
Okay.
going through the species one by one, and then we can take those questions on invasives.
Okay, let's
That's
do
right.
that.
Okay.
And then one question about the burrowing crayfish, and then we
Okay.
can go on.
Sure.
How do they do anything special to keep their burrows from caving in?
Well, yes and no. The yes is they tend to select a soil type that's got a little bit of clay in it that allows those burrows to kind of firm up a bit. You won't find burrowing crayfish in soil types that are really, really sandy, because that soil type will, as I mentioned, collapse on itself. So by sticking to a soil type that has more clays to it or firmer soil, those burrows do stay open and functional.
All right, thanks.
Okay, so we have an anatomy lesson before we start talking about identification. Unfortunately, crayfish aren't like birds where you can spot them with binoculars from 10 to 20 yards away and look at distinct color patterns to do identifications. And so with this slide, I just want to introduce you into a few structures that we're going to be talking about. And then structures we're going to talk about when we talk about the life history of crayfish. Rostrum, as I already mentioned, is this kind of spiky, in not all species, but some species, a flat structure that's between the eyes and extends out a bit. The areola is that structure I mentioned where these gill chambers are down underneath, and those chambers are demarcated by a very slight ridge or line that you can see with your naked eye in the back of the crayfish thorax. So there's a line that kind of bends at an arc here and another one here. This is what we call the areola. The chele is another name for the claw. And the claw is the highly modified first pair of walking legs that are called pariapods. So start at the front. 1, 2, 3, 4, 5 pairs of walking legs. The first one has been modified, highly modified to have that claw, which they normally use for defense, not predation, not grabbing prey items. Then they have structures called the pleopods or swimmerets. And again, there's 5 pairs of them. The first one is here. You see the Roman numeral 1 within the blue circle. Then there's 2, 3, 4, and 5. Here's the 10 pairs of non-feeding appendages that give them a name and allow them to make them part of Decapoda. These first pair of pleopods are also called gonopods and are found only in the males. And the reason I'm pointing this out is, unfortunately, to do a really accurate identification, it's going to come down to, in a few cases, to looking at these male genitalia and their shape and size to do proper identification. All right. So again, that's what I mentioned. Those first pair of chele or claws are the first pair of walking legs, even though they don't use them for walking. All right. Just some actual photographs here. Here's those first pleopods or gonopods. Here's the last pair, number 5 of the walking legs. These gonopods are the male sperm transfer organs that lay between the bases of these latter 3 pairs of walking legs. And usually, sometimes they can be very hard to see. You see a lot of little short setae here or hairs. They're laying down in between. Again, these are what the male uses to transfer sperm to the female. If you use a knife blade or a small probe, you have to lift those structures up and out of this kind of groove they're sitting in, and that these gonopods have been lifted out, and that's what we're seeing here in this image on the lower one. Okay, and there are a pair of them. This is the first pleopod or gonopods. Females just have a sperm receptacle organ. This is called the annulus ventralis. So crayfishes are very easy to sex just by flipping over. This is a structure that the male transfers the sperm in. The female can store that sperm for months and months and months and still keep it viable, and then we'll extrude it and combine it with her eggs that come out the base of the third pair of walking legs here, this female gonopore. She'll release the eggs. The sperm has been stored in the storage sac. They'll mix. The eggs will be fertilized, and then the eggs will move back to the underside of the abdomen where she'll hold them until they're ready to hatch. Okay, so that family Cambarity, this illustration is the best out there, but it can kind of make you go goofy trying to figure out what's going on with it. The big thing for identification of crayfishes in the family Cambarity is this form alteration between form 1 and form 2. Form 1 are males that are reproductively mature. Form 2 are reproductively immature. And if we look at this illustration, this is kind of a basic life cycle of a Midwestern crayfish. And if we start right here at this middle point, this is when the eggs hatch. And it's normally, might be February, it might be March over here. And if we start here and start moving, here's where it gets hard. If you start moving out right through here, going through the first year as a juvenile, because that line is white, it means it's in a form 1. So that white concentric band coming out means the juvenile is form 1. It's going through that first year of life. And then it gets to the point right here where it turns to a gray shading in September, usually in the fall. Since it's an arthropod, like an insect, it has to molt, and it molts its skin, and it molts, it molts into the form 1 stage right here. And then as we're moving around, it stays as a form 1 through the winter and spring months. It molts back to the form 2. Usually this is May, goes through the summer as a form 2, then molts back to the form 1. Most copulation occurs in the fall months. So that's when that male is a form 1, and then it molts into the form 2 during the summer months. Okay. Usually in the spring months, March, April, over here is when the female again will extrude her unfertilized eggs, combine it with the sperm that she's been storing in that annulus ventralis. The gametes will combine. She'll move those eggs, fertilized eggs back. She'll hold them with a light glue substance under her abdomen. The eggs will hatch, as you can see in this lower image. You see little tiny black flecks. Those are the eyes of the juvenile crayfish. So they'll hatch into a juvenile, stay attached for a week to 10 days, maybe 2 weeks, and then release and start life on their own. The form 1, the molt from the form 1 to the form 2 is important to mention because we usually have to have that form 1, that reproductively mature male, to make identifications on lots of species. Not so much for Wisconsin because the diversity is lower, but in other states we need to have those form 1s. This illustration just shows you the difference between a form 1 on the left and a form 2 gonopod on crayfishes. The appendage or the element becomes much more sharp, much more pointed, and you'll see some dark yellowish, what we call sclerotization. This is a different genus over here. Instead of being kind of a uniformly colored, rounded element up here, it becomes much more defined with a sharper tip. Same thing down here. You see these elements. This is a Procamberous species where there's 1, 2, 3, 4 different elements. Here's one. Here's a form 1 again on the left versus form 2, where it becomes much more sharply pointed, and you'll see that kind of dirty, dingy yellow color to it, like you see on these lower 2 examples. Okay. The number, size, and shape of these elements is what we use for identification for lots of species. Okay, so who lives in Wisconsin? I'm going to try to go through this quickly. There are 9 species. This is a list of those 9. The 2 that are invasive are listed in red. To do an identification, I don't know how many of you have used the dichotomous key. That's usually the best way to do it. This is just an example on the right here. You just go through a system of checks by looking at the morphology of the crayfish you have in your hand, and then does it have character state A or character state B, and start moving down that flow chart to eventually get at the species. Again, this isn't for Wisconsin. This is just an example of how that dichotomous key works downstream to arrive at an identification. So for Wisconsin species, the first characteristic that you would need to look at is this areola structure. Is it linear? And that means do those 2 lines come together and touch? Or is it open? And open can be just very narrowly open or widely open. You can see that gap between those 2 lines. Okay, so let's start with linear. If you collect a crayfish, you look at it and you see that these lines come together and touch, you're going to go down the path to the left. And then the next character you would look at would be the palm of the chele. Does that palm have really sharp, almost like sawtooth-like, what we call tubercles or spines along the opposable margin? So this image is on the right. If you were looking down at a crayfish, this would be on the chele or claw that's on the right hand. It's the movable finger here shows you that it's the right chele. So I've highlighted these very sharp sawtooth edge tubercles or spines. If it has that, you go then in direction A. If you just see lower rounded bumps along that margin, then you would go B. If we go A, that is this image is here by mistake. Okay, so let me back out of my presentation and get rid of that slide. Okay. All right. So we're going with spines or sharp sawtooth edges here on the palm. The next character you're going to look at is the rostrum. Is that rostrum really strongly tapered and have very sharp, small spines at the base of the central spine? If it's yes, and the body is also covered by small bumps, it's the red swamp crawfish. Now that's a typo. It should be crawfish, not crayfish. If the rostrum does not have those sharp spines, little tiny spines here, if the margins of that rostrum kind of smoothly round in, come down in a slope, and then round to the edges, and the body is not covered by bumps, it's the prairie crayfish. Okay, so if it's the red swamp crayfish, this is one of the invasives. Here's a couple images. Here's the range in Wisconsin. There's usually 1 or 2 sites in the Milwaukee area so far that it's been recorded from. This is an example of why we can't use color for identification of a lot of crayfishes, because you can give a really deeply red color in red swamp crayfish. That's where it gets its name, red swamp. But it's not always there. Sometimes you can have some individuals that might have some lightish, brownish, yellowish colors. So life would be easy if there was a distinct color pattern. Again, I'll point to birds. Birds have very distinct color patterns. that allow for identification. For some crayfish species, there is some variation as the individual gets older. In this case, in the red swamp crayfish, that red color becomes more obvious, okay? So if you made it to this point in the key, you've likely got a red swamp crawfish. Okay, if you went the other direction with smoothly rounded rostral margins and no bumps, it's the prairie crayfish. An example here of a color photo. It's usually brown, sometimes orangish. If you'll notice from this figure, it's kind of restricted to the southeastern quarter, extreme southeastern quarter of Wisconsin. So not a wide-ranging species in the state. All right, so if we go back to B, and the claws did not have those sawtooth edges. They were very small bumps. Plus, again, the linear areola. You have two choices. Is there a movable, does the movable finger have this cleft, this deep incision into that movable finger? If the answer is yes, it's the digger crayfish. If the answer is no, there's a little bit of an incision here, but it's smoothly or moderately concave. There's no deep cleft right here at the top, okay? If it does not have that, it's the Great Plains mudbug. Here's the prairie crayfish. Again, there's a little hint of gray here in the southeastern portion of Wisconsin, again, where you find prairie crayfish. So again, not a very widespread species in the state. Sorry, the digger crayfish. All right. If it did not have that deep cleft, it's the Great Plains mudbug, pretty much statewide in Wisconsin. Okay. Another thing that sometimes you'll see in the Great Plains mudbug is this kind of light colored stripe down the middle of the tail. This is a primary burrower. This is a primary burrower. And the prairie crayfish from the previous slide are all primary burrowers. So you might see these guys out walking around on spring days when we've had heavy rain, but outside of that, you will usually not encounter these primary burrowers in a stream or a lake. All right, so back to the original character. Let's say the areola was open or linear. And again, it can be narrowly open or very wide. The next character to look at, you can kind of use red color. That's why I use the word usually, because it have very narrow claws and 3 or more elements of the gonopod. So here we're getting into that male characteristic that unfortunately you're going to need a male for proper identification. You can see in this photograph, there's 1, 2, 3, 4 different small elements at the end of that gonopod. If there are 3 or more, it's the white river crawfish. If it's not red in color, if the claws are wider and there's only 2 elements, only 2, it's going to be several different species. And notice the variation here. We'll cover straight elements versus curved elements. Okay, so the white river crawfish, fairly widespread in Wisconsin. You see in the map in the lower right, most of the southern and central portions. It does get color, very similar to the red swamp crayfish, but the way you'd separate a white river crawfish from a red swamp crawfish is, again, going through that first couplet, noticing that these lines on the back come together and touch, or if they're open enough for a space. You can see that a little bit here in this photograph on the left. And again, color can be variable. As they get very large and mature, maybe a 3-year-old individual will get that deep red color, but something that's a year and a half old, it might be more brownish. Okay, so if there's only the 2 elements, you got to look at if the elements are curved versus straight to determine which way you go. If they're curved, are they curved and shorter? If that's the case, it's the calico crayfish. If they're curved but longer, both elements arch over slightly, much, much longer. So again, if you look at kind of the length of this element versus the whole length of this structure, it's maybe about half the length and curve is the calico crayfish. If the length of these elements, so from here to here, is much, much greater than the total length of the structure, it's the virile crayfish. Okay, this is the calico crayfish, again, another species in the extreme southeastern portion of the state. Calico crayfish generally also have a deep incision, like the digger crayfish, but again, it's going to have an open areola, not a closed areola like the digger crayfish. The virile crayfish, one of the most, probably the widest ranging species in North America, goes up to the Hudson Bay, down to extreme southern Illinois. Longer and curved. You'll sometimes also see these kind of paired dark marks on each abdominal segment. And sometimes you'll see, well, most times on larger individuals, you'll see these cream colored tubercles or bumps along the edge of the claws. And notice the blue color here. Sometimes individuals of several different species of crayfish that don't get a lot of sunlight will get that blue coloration. So again, using just color alone is not that useful for identification. All right, so let's say the elements were straight. Are they straight and long versus straight and short? And does the element have a shoulder, an angular shoulder at the base of those elements? If it looks like this is the Form 1 and the Form 2 gonopod. It's the rusty crayfish. If it has a much shorter but two straight elements with no angular shoulder here, it's the northern clearwater crayfish. Rusty crayfish, we mentioned. You'll see the invasive range in orange. So you can tell from image quite quickly that thing is spread widely across the Midwest. They get their name from this rust-colored spot on the side. If you look at this central image, most individuals are easy to identify from that rust-colored spot. However, I've got this picture on the left to show you that sometimes that rust-colored spot can kind of blend in with a little bit of darker coloration on the top of the animal. So sometimes it's not that easy to see. It is there, just not as obvious as it is here in the central image. Okay, the last species is northern clearwater. Again, if they were shorter elements that are straight, no angular shoulder here. It's the northern clearwater, another statewide species in Wisconsin. This is the species that's been displaced in lots of locations by the rusty crayfish. There's usually a dark wedge that I've noted here on all three images that run down from the back edge of the thorax all the way down to the abdomen, this tapering dark wedge shape that you see here. Okay, so looks like I'm close, couple minutes over. I just want to share a couple of resources with you to use that can be useful for identification. A lot of this data on distribution in Wisconsin crayfish comes from a publication the Milwaukee Public Museum did in 1988. It's Hobbs and Jass's The Crayfishes and Shrimps of Wisconsin. I've not been able to find a free download PDF of this publication. There are used hard copies available for sale. There's lots of background information on the biology of each of these individual species in Wisconsin, but being from 1988, some of that distribution data has changed, obviously. So it's a little bit dated, but still some really good information on biology of them. I don't make money on the sale of this at all, but we did do a field guide to the Midwest. Most of those range maps and a lot of those photos I've used in the preceding slides came from this field guide to crayfishes of the Midwest. This should be 2015, not 2915. So 2015, and that did not get checked. That is, hold on, let me fix that because this is going to go record. So it's an Illinois Natural History Survey publication, manual number 15. You can buy hard copies on Amazon or through the Prairie Research Institute, my home institution, at this website. If you're interested. Again, I don't make money on it, so I'm not trying to hawk books here. We don't have a digital version yet. Okay, one last resource that can help with identification of crayfishes is kind of knowing what's around. If you find a crayfish in your area, it's useful to go to a website and look at what's been recorded from your area to give you a starting point. It's not the final say, and the reason it's not the final say is, as we've seen from rusty crayfish and red swamp crawfish, new species are getting moved around continuously. So just because there's no dot for a species in your location, that doesn't mean you may not have unfortunately picked up a red swamp crayfish. So we put together a American Crayfish Atlas that allows you to plot the location of any and all species that have been stored in major museum collections. And I'm going to try to exit out real quick and just show you how to work it. We're having some update issues on Google Maps. So it does normally show up better, but unfortunately, the update issues started developing yesterday. Okay. Are you guys seeing all of my screen? You can see this?
Yeah, we see everything. We see your desktop and there you
Okay.
go. Perfect. Now it's
So
just
let's,
that screen.
when you go to this website, if you just go to you can look at species by state. This will give you a rundown of every state and the species that have been recorded in those states. But you can also go to the map feature. And again, this is blacked out. We're sorry, it's darkened here. Google's having some update issues. You can go over here to the upper right corner and click all species, and it will plot every record of every crayfish. And what that would allow you to do then is there should be a zoom bar here. You can start to here's Kansas City, but we'll scroll up here. This can be helpful for identifications also. So you'll see all these different colored dots, and the different colors mean you can click on it and it will tell you what species. So that's the virile crayfish. The blue dots are the northern clearwater crayfish, for example. So it can be very, very helpful just to know what's around. If you don't have a key or you don't have, I'm guessing you guys will have access to this PowerPoint. Correct? That you can save the
Oh, we
links
have the
for?
recording. The recording
Okay.
will be stored.
Okay. So if the recording is stored, you'll have access to this key I put together for the talk to help you if you're, really eager and you have a crayfish in your hand and you have access to this to start working it through the key, or you have that like the field guide for identification. So anyways, okay. That's my last slide. I'll be happy to thank you for your attention, and I'm glad to ask any questions or answer any questions.
Yeah, we have quite a few questions come in, so we're definitely going to go past 1 o'clock. But I just want to acknowledge for everyone if they have to run at 1, you can always hear these questions and the answers on the recording. So to start, there's one question that got a lot of votes. And it's which southeast Wisconsin populations of red swamp crayfish are you referring to? This person's saying that red swamp crayfish were not established in Wisconsin currently. There was a population in Germantown, Kenosha area in the early 2010s that was eradicated.
Yeah, well, that's basically I thought there were 2 locations in 2 different ponds. And I knew there was some pretty intensive eradication efforts. I have not seen that those were entirely successful. So that could well be the case that they have been completely eliminated. I don't know that I have heard that. Getting rid of anything, I've seen lots of talks at scientific conferences from Michigan, where they're having the same issue that red swamp crayfish got introduced into some of these ponds at corporate headquarters. One may have been a golf course. And they're working really hard to get rid of them, but getting 100% eradication is a tall order. So I hope you're right, whoever asked that question. I hope they have been completely eradicated. I just had not heard that those eradication efforts have been 100% successful.
Awesome. And Maureen, go ahead and ask
Okay,
your question live.
Yeah, I saw you unmuted and I wasn't sure what to do. I didn't ask the question, but I was one of the people who liked it. But actually I went into our database. So I'm Maureen Kelcher. I'm our statewide aquatic invasive species monitoring
Okay.
coordinator. So I provide guidance to the DNR staff to look for things, report them, and update like our website. And I was going to add a comment on that like, no, we have them on our website. Here's a link. And like, we don't have it. We haven't properly tracked the species on our website. So you wouldn't have a way to know that we don't see them anymore. We haven't seen them probably since 2012, maybe 2013. So it's been a long time, and we should have updated their status to be no longer observed. There was quite an extensive effort to fill some of the ponds and then to control it. There's no populations that we know of anymore in southeast Wisconsin. There is one population that was observed along the Wisconsin River, and we haven't seen that for a few years, but it's still listed on our website as verified. But that's the only population, but we haven't seen it since it was first observed.
All right, that's great.
Yeah. I'm glad that this was brought up, because now we're like, oh my God, we have to clean our data better. So it's a bit embarrassing.
So I mean that's basically the record I was referring to. I was kind of involved with some emails back and forth, and did you guys partner with Auburn University to look at using CO2 and things to get rid of them, or did you use proton?
I wasn't in this position at that time, and I don't know if Scott Venegrin is on the call or Heidi Bunk. They would be able to speak to it a bit more. There could be another person, but we used a lot of chemicals in the water body. It was a massive and very expensive effort.
Well, I hope it's successful, because as I said, it's because red swamp crayfish can also burrow a bit. They've shown a resistant. They'll dig a burrow into the stream bank. If the DO and the water gets so low, they'll crawl out of the pond and then dig into a burrow, and are shown some tenacity for surviving.
Yeah, I think it might have been 2013 or 14. It was a very decrepit crayfish, but just a single one had been observed. But I think it was $1 million effort. Like, it took a lot. Yeah. Wow. We got a big grant.
Yes, I want to make sure we move on to the other questions, but it sounds like the populations were eradicated in those ponds. Scott submitted a few comments related to that too. So a few other questions as well. One is an easy one. Where did rescue crayfish originate from?
Yeah, okay. So if you go back can you guys still see part? Let's go back. Do you see my PowerPoint, just not on the full screen?
Yeah, it's very tiny though. It's very hard to see.
Let me go full screen quick. You see the gray region here?
Yes.
That's their native range. So basically, the middle Ohio River, there also occurred. Our field guide didn't cover Kentucky. It was just the upper Midwest. So they were native to the northern portion of central and eastern Kentucky, and then in the Ohio River drainage, right on both sides of the Indiana-Ohio border. The Maumee River flows into the eastern Lake Erie. It's debatable if they're native to that portion of Lake Michigan, because they built a canal connecting the Miami and the Maumee rivers in the late 1800s, I believe, and that could have allowed the rusty crayfish to move up in there. So it's unsure if they're native to the western Lake Erie, but that's where they came from fishing bait mostly. They somehow got into a few bait dealers that must have been based somewhere there in the Ohio River drainage, and shipped those bait individuals across the Midwest.
All right. There is also a question related to how do these invasive crayfish impact the burrowing species, or are they not overlapping in habitat?
We have not seen that yet, because there's been no case of an invasive primary burrowing crayfish. So we don't know. Okay. Everything that's been invasive to this point has been a non-burrowing crayfish. Again, even though red swamp crawfish can dig a burrow, I don't consider them to be a primary burrower, more of a secondary burrower. So the primary burrowers have not been impacted yet by any invasive species.
And that's why the northern Clearwater, Wisconsin species is being affected by rusty because they're overlapping in their
Yeah, yeah,
non-burrowing habitat.
correct. Yeah, they share almost the same habitat type under
Okay.
rocks and gravel um, on the bottom substrate.
All right. We had a few people who must be our lake monitoring volunteers, perhaps. They are seeing signs of diminishing populations of rusty
Yep,
crayfish in their lakes, Vilas County lakes up north in northern
yep,
Wisconsin. We have Crescent Lake in northern Wisconsin as well. And one person said they heard potentially it was liver virus and they disappeared. So yeah, any comments about that?
yeah. So yeah, I've seen the same thing. Dr. Eric Larson's here on this campus. He's kind of taken over some of the crayfish work up there at that Lake Biological Station in Vilas County, and they are definitely seeing some reductions in rusty crayfish populations. One of the theories is that liver virus, but I think it's still a bit early to know for sure if that's the primary driver. It is a very good sign that we're starting to see reductions in rusty crayfish in some of those lakes for sure. Is it the typical boom and then bust population that we see in other aquatic invasive species? Hopefully, that's the case. And I have seen some locations, not all, but a few locations in Illinois where it was just northern Clearwater crayfish, and then it was just rusty crayfish, and now we're starting to see a few individuals of northern Clearwater crayfish start to show up. So hopefully it's that boom and bust cycle that we're starting to see other natural biological control mechanisms, i.e. predators are starting to get better at foraging for rusty crayfish. That's having an impact on those populations. So yes, yeah, that is an encouraging sign that some of those lakes are starting to see a decline.
Awesome. Someone also asked about the devil crayfish. And is this person thought they were in Door County. Um, do you know anything about that?
Yep. Okay. So I forgot to mention, I was trying to move a little too quickly. Let's go back to this slide. So you'll see the Great Plains mudbug here. And the second species listed, and then parens, I've got formerly devil crayfish Cambris diogenes. And so what's happened to that devil crayfish Cambris diogenes? You will see that in the literature. It's a taxonomic change that's happened in the last 15 to 20 years. And I've marked on some of the Faxonia species down here. I'll have formerly Orconectes propinquus, formerly Orconectes immunis, formerly Orconectes rusticus. That's a taxonomic change that's occurred. It's one and the same species, but some DNA work has said that almost everything that was in the genus Orconectes is now in the genus Faxonius. Okay. But back to the question about devil crayfish. The devil crayfish was a really wide-ranging single species for almost 100 years, everything from southern Ontario all the way down to Texas. And as taxonomists and researchers have started to look closely at the DNA variation within that formerly really wide-ranging species, they're realizing that there are unique lineages with unique morphological and genetic characters, and they're splitting those out into different species. And so everything in Wisconsin that was formerly part of that large single species, the devil crayfish, Cambris diogenes, is now what we recognize as Lacuna cambaris, a different genus, nebraskaensis. So the devil crayfish name is still out there, but that group has been split into multiple species, and what's in Wisconsin now is not the devil crayfish. It's this Lacuna cambaris nebraskaensis. Does that make sense?
That makes sense to me. Hopefully, it makes sense to the person who asked the question. Really interesting.
It's just new data. I mean, new science coming in that shows that the taxonomy needs to be adjusted. Taxonomic names are not stable.
Hmm.
New data requires changes to how we name and organize these things.
And I think this might be related, but someone had asked what the crayfish species is associated with the Heinz emerald dragonflies. Is that this one?
Yeah, it's what used to be the double crayfish, so Diogenes. Yeah, the primary burrowers at least in Wisconsin, would be Lacuna Cambarus nebraskaensis now. Yeah, for sure.
Okay. So we just have two more questions.
Okay.
I'm just going to read this. What is your recommendation to survey and conserve populations of primary burrows throughout wetland scrape creation? Additionally, are chimney surveys a good way to quickly assess population density? Any suggestions on survey methodology would be appreciated.
Yep. It's
That's a
exactly what.
complicated one.
No,
If that requires an email response, let us
no,
know.
no, but it's my lab has been working on this now for the last 5 or 6 years. Um, how is wetland restoration? We're just starting a new project in Missouri that's going to look at what kinds of specific wetland restoration activities, how those impact burrowing crayfish populations. So the first part of the question, and this is from papers that some of my former students have just published, as long as the soil characteristics and the water table depth can be maintained at a more normal level, these primary burrowing crayfish seem to be able to do okay. We've done some modeling that shows that the vegetation isn't critically important. The canopy cover isn't critically important. It's just the soil type, and more importantly, where the water table sits in that wetland area that would assist with burrowing crayfish. Burrowing crayfish are pretty hardy organisms. It takes an awful lot to really have a detrimental impact. We find them in roadside ditches that are mowed twice a week. They seem to prefer roadside ditches in front of Days Inn hotels or Subway restaurants, completely denuded of everything but grass. But if the soil type and the water table is suitable, they'll live there okay. So maintaining the water table is probably what I think right now, what the data suggests is the most important aspect. The second part of the question was counting burrows. Yes, but a better way to do it is counting what we call active burrows, because a lot of these burrows can be abandoned. And when you go in this, the best way to survey for active burrows, that's a burrow that's inhabited by a burrowing crayfish, because they can abandon some of their burrows. Is after a rain event to go out and look for signs of fresh mud that's been pushed out of that burrow opening. Rainwater and mud will get washed in during a rain event, and those crayfish, they're just like beavers. They're working nonstop. They're really industrious organisms, and they'll start pushing that water and mud that's come down into their burrow out. So you can walk along and look at burrows and see a lighter shaded mud that's a little bit slick. You can figure it out pretty quickly that something's been pushed out of that burrow within the last 24 hours, and look for evidence of like cobwebs or debris that's fallen into a burrow opening. If you see that kind of stuff, it's likely that there's nobody home in that burrow. If you don't see any debris or cobwebs and there's signs of fresh mud, you can be pretty certain that there's a burrower in there, and that would probably be a more accurate count of density or population size in a given area.
That is fascinating. All right, so last question. I think this is a good one to end on because it's related to climate change and the future for crayfish. So is the spread of invasives, this is kind of the first part, is the spread of invasives at all associated with range expansion due to, our changing climate perhaps? And what are your thoughts on native species expanding their ranges as a result of climate change?
I think it's too early to know for sure. Um, I have not seen any data. It's not the kind of research that I've been conducting. There's been some modeling, some predictive modeling that uses temperature. You measure the temperature and the native range of a species and look at these different climate change models and project that on a map. It can give you some insight that some of these species might be moving farther north. I do think that's probably what's going on in the red swamp crawfish. They are abundant, uh, abundant in the Chicago River drainage in Illinois. And the northern extent of their range used to be the extreme, their total global range. Red swamp crayfish came just to the extreme southern tip of Illinois where it borders Missouri and Kentucky. And the climate difference between extreme southern Illinois and Lake County, northern suburbs, or downtown Chicago, it's, I just looked this up a while ago for another presentation. It's 7 or 8 degree difference, average temperature for winter temps. And now we've seen red swamp crayfish definitely established in these waterways in the Chicago region. I would speculate that temperature, we're definitely having milder winters here. Probably has had an impact on red swamp crayfish being able to do quite well in the Chicago area when historically, it might have been just too cold for them. Otherwise, they've had 10,000 years to move north in Illinois since the last glacial advance, and they haven't. But now that they get moved up by humans and they become established, I would say that the slight temperature increases we've seen hasn't hurt them. Has probably helped.
Okay.
That's the only example I can think of that's some direct, I'll say some indirect evidence that climate change will have an impact on crayfishes. There just aren't any other examples that I'm familiar with that shows a continuous spread north in the last couple of decades. And part of the reason for that is there just aren't that many people that are working with crayfish. There's a lot more biologists that are working with fish and can see some possible range changes, but we've been hindered by a lack of historical distribution records for crayfish, because when I first started working on crayfish in the early 1990s, I could count on one hand the number of researchers that were doing anything with crayfish in the whole US. And that contributes to this lack of baseline historical distribution data that makes it more challenging to look at distributional changes for crayfish.
Thank you so much for that. Yeah, maybe this will inspire someone here to, get more involved in collecting data on crayfish, perhaps submitting to iNaturalist, or getting youth in their life interested in crayfish. So thank you
They're
so
very
much.
cool animals. People say, how'd you get into crayfish? I remember as a kid going out to a stream and collecting one in a cup. And I just thought it was the coolest looking animal I'd ever seen. Took it home and put it in my aquarium. And, just led to, I guess, this career. So they are just really, really interesting organisms for several different regions.
Yeah. Well, Paul, you want to wrap us up?
Yeah, Chris, we had over 100 people here at one time, so there's a lot of interest in these little
Oh,
critters.
awesome. That's awesome. I hope.
Yeah.
Wisconsin has had a pretty good history of paying attention to crayfish. Unfortunately, it's mostly due to the invasive issues. Back in the mid 1990s when they started, mid 1980s, when it was becoming obvious what rusty crayfish were doing in northern Wisconsin. So I hope more people start looking for burrowers. They're understudied because they're just hard to collect. But there's an awful lot to learn about the biology, diet of primary burrowing crayfish.
Well, thanks so much for your presentation. It was very interesting, and I think we see a lot of hearts and clapping hands
Lots
moving
of things
across the
floating
screen
around.
here. So people enjoyed it. Thanks a lot, Chris. Thanks for everybody for attending, and hopefully we'll see you next year for our winter water talks.
All right. Good deal. Thanks, everyone.